Krebs Cycle. It was named after the Hans Adolf Krebs, who discovered it in 1937. It is also known by several other names: the citric acid cycle and the tricarboxylic acid cycle. This cycle occurs in the mitochondrial matrix. It is the series of biochemical reactions in which the acylated portion of acylated-CoA is oxidized to carbon dioxide, and the reduced coenzymes FADH2 and NADH are produced. The Krebs cycle is what is known as amphibolic in that it is both catabolic, breaks down molecules, and anabolic, builds molecules. It is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate derived from carbohydrates, fats, and proteins into carbon dioxide. Each stage in the cycle and in the link reaction, pyruvate conversion into acylated-CoA occurs twice for every glucose molecule that enters glycolysis, because 2 pyruvate molecules are produced for each glucose. The 8 steps of the citric are a series of redox dehydration, hydration, and decarboxylation reactions. Each turn of the cycle forms 1 GTP or ATP as well as 3 NADH molecules and 1 FADH2 molecule, which will be used in further step of cellular respiration to produce ATP for the cell. Oxidation. Removal of electrons from a molecule. This subsequently lowers the energy content of a molecule. Most biological oxidations involve the loss of hydrogen atoms. This type of oxidation is referred to as dehydrogenation. The enzymes that catalyzes these reactions are called dehydrogenases. Gain of oxygen atom. Reduction. Opposite of oxidation, it is the addition of electrons to a molecule. When a molecule is oxidized, the liberated hydride ions do not remain free in the cell. In order to harness the energy of these electrons, they are immediately transferred to another compound by coenzymes, or loss of oxygen atom. Phosphorylation. Accomplished by transferring a phosphate group to ADP. Decarboxylation. Carbon chain is shortened by the removal of carbon atom. Isomerization is the process by which one molecule is transformed into another molecule which has exactly the same atoms. Dehydration, removing of water molecules. Hydration, addition of water molecules. Reactions of the citric acid cycle. Step 1. Formation of citrate. Acylated-CoA, which carries the 2-carbon degradation product of glycolysis, enters the cycle by combining with the oxaloacetate to give citral-CoA. The addition is catalyzed by the citrate synthase. Citral-CoA is hydrolyzed to citrate catalyzed by the same citrate synthase to produce CoA and citrate. Step 2. Formation of isocitrate. Citrate, a tertiary alcohol, is converted into its isomer isocitrate as a secondary alcohol in an isomerization process that involves dehydration followed by hydration that are both catalyzed by the enzyme aconitase. Step 3. Oxidation of isocitrate and formation of CO2. This step involves oxidation reduction, the first of 4 redox reactions, the Krebs cycle, and decarboxylation. Step 4. Oxidation of a ketoglutarate and formation of CO2. This second redox reaction of the cycle involves 1 molecule each of NAD plus CoA and a ketoglutarate. Step 5. Thioester bond cleavage in succinyl-CoA and phosphorylation of GDP. 2 molecules react with succinyl-CoA, a molecule of GDP similar to ADP, and a free phosphate group. Step 6. Oxidation of succinate. This is the third redox reaction of the cycle. Step 7. Hydration of fumarate. The enzyme fumarate catalyzes the addition of water to the double bond of fumarate. Last is step 8. Oxidation of L-malate to regenerate oxaloacetate. This is the fourth redox reaction of the cycle.